Hey there, pre-AP. We're almost done with our biochemistry unit, so we're on to our last outline, 2.5, metabolism and enzymes. So have your highlighter, have your pen ready, your pencil, so that we can annotate, make notes on these things, highlight what's important, and draw some pictures that will help us. So please highlight that what metabolism is. All metabolism is the sum of all the chemical reactions occurring in an organism. Constantly, your body is either building up molecules or breaking them down. So let's talk about our first type of metabolism. It is catabolism. Catabolism means to break something apart. That would be if we had, put yourself out an example here, if we had salt, sodium chloride, plus, let's say that we put it in water. Think about the type of bond that holds salt together. Is it covalent, ionic? Very nice, it's ionic. And what happens to ionic bonds whenever they're placed in water? That's right, they break up. So, remember your arrow means something happened, and it turned back into just sodium ions and chlorine ions. That's catabolism. We break something down. Please know that because we're breaking something down, we're releasing that potential energy that is carried between that bond that's holding sodium and chloride together. Because we're releasing potential energy, we say that it is exergonic. This is an exergonic reaction, which means as these break apart, that heat is going to be released. So not only are we going to have sodium and chlorine as products, but heat will be as well because it's going to be given off into the atmosphere. But let's talk about our second one. It's the opposite of catabolism. It is anabolism. Anabolism means that we're building something up. We are requiring that kinetic energy, that energy of movement, to be able to create a new chemical bond. So let's say that we had, let's think about this. It says that we are going to build some bonds between monomers. Think about your monomers that we talked about. One of your monomers for carbohydrates is glucose. We're gonna draw the chemical formula for glucose. Hold on, let me back that up. I misspelled glucose, oh my goodness. Glucose plus glucose equals sucrose. This is a one monomer plus one monomer equals a polymer. Two or more molecules, two or more monomers coming together. So this is building up a reaction. We are building a bigger molecule called sucrose. And we say that this is an endergonic reaction. So please highlight that because it absorbs energy. You have to have heat to add these two things together. And think about, to tell the difference between catabolytic and anabolytic metabolism, look at C below catabolism and the C below anabolism. I think of catabolism like a catastrophe. Something bad has happened. If there's a catastrophe, usually things are falling apart, like buildings and an earthquake. Or if you're anabolytic, you're building something up. So think about anabolic steroids. Think about steroids help your muscles. Most people take those to build their muscle up. So think about it like that. If we think about energy, moving on down our outline. Energy, remember there's kinetic energy and potential. Remember, kinetic is the energy of movement. Potential is the energy of position. And remember, whenever we're talking about potential energy, highlight that it's stuck in between those bonds. Like that sodium and that chlorine, there's a bond between them. It is an ionic bond. And because there's an ionic bond, it's storing energy. So please also highlight for living things, the chemical energy in life is found in chemical bonds. Now let's look at our next part called thermodynamics. So all thermodynamics means thermodynamics, please highlight, is the study of heat. So think about thermo. Thermo means heat. Temperature. You use a thermometer to find your temperature, how much body heat you have. So we have two laws. The first law is called the principle of conservation of energy. Please highlight, all it means is energy cannot be created nor destroyed. It's only transferred from one form to another. And then our second law is called entropy. Entropy, all entropy means is that means a disorder that causes something to be unable to work because there's a low state of order. Now, whoa, what does that mean? Look at the number 1 above it. This means that energy transfers increases the entropy of the universe. Now you're like, well, I still don't understand, Miss Minor. We'll go down to the number 2. Sunlight. Remember, sunlight has energy in it. It's going in and heating up something. It's coming out of a reaction. Think about that exergonic reaction. Because it's leaving that reaction, it can't do work. We'll try to demonstrate this in class. And then we look at Roman numeral 4, Gibbs free energy. Please highlight that it's the free energy just hanging around. Free energy just hanging around, waiting to do some work on something. Build something up, break something down. We mainly do this by making either ATP or GTP. ATP is going to be our main guy. GTP has a little bit of energy, but not as much as ATP. Because remember, like it says in big letter B, not all energy is available. Some can be lost as waste whenever you release that energy, that heat energy, from an exergonic reaction like breaking up that salt. And remember, just like we just said, that number 1, most of your energy is lost as heat because it's a byproduct of most reactions. We're going to see that in photosynthesis and cellular respiration. So the types of work that can be performed. Types of work, you can be mechanical, which just highlight, it's outside of the cell. Maybe you're fighting off invaders today. Transport across the membrane, that takes energy. Remember, active transport takes ATP to send things from a low concentration to a high concentration. And then metabolic. You're either building something up, anabolism, or breaking something down, catabolism. ATP, oh my goodness, put a big O star by ATP. That stands for adenosine triphosphate. Let's draw this guy out. Adenosine is one of those lovely nucleic acids, so we're going to give him the letter A in a box. He's a different thing, so he gets a different shape. And we are going to draw 3 phosphates. Think about the prefix tri. If you need to write that down, it means 3. So this is the adenine, the A, and 3 phosphates. Now, this adenine is made of ribose sugar. We'll get more into that whenever we get to DNA. And so those phosphates, it says there's 3 negative phosphates, so give them a negative sign. And because those 3 phosphates are sitting beside each other, I like to think of them as 3 little kids sitting on a bus seat. If you've ever had to sit 3 people to a bus seat, that is crowded. Oh my goodness, you don't want to sit crowded 3 people to a seat on a bus seat. At some point, if you really don't like the third person sitting with you, if you want to be mean, which is not, don't be mean, but some people are mean, and they're going to kick the third person off the bus seat. So, let's highlight this, what I just said for letter B. There are 3 negative phosphates linked together, and they are highly unstable. Unstable means those 3 phosphates are mad at each other. There's 3 kids on a bus seat. There's no room. So, because of this, it being unstable can kind of be a good thing with molecules because it has the capacity to do work by breaking one of these bonds between the phosphates. So, if we kick this phosphate off the bus, it's going to now turn into a different molecule called ADP, adenosine diphosphate. So let's highlight that on big letter C. ATP converting to ADP gives off energy because we kicked that third phosphate kid off the bus seat. And because we kicked him off, we broke a bond in here. And remember, when you break a bond, it releases energy. So, later on it says, in a little bitty parenthesis, ADP, this guy, will be recycled back into this in some chemical processes. And we'll find out those later on. And then our next thing, this is about, let me change colors. Phosphorylation. That's a very important process. You have to know phosphorylation for a lot of things. Please highlight that it's the attaching of an unstable phosphorus ion to another molecule to make it unstable, thereby able to do work. So this would be your example. Draw this out there. This would be like ADP, adenosine diphosphate. Let's say that you added a P. Because you're adding that P to the molecule, that's phosphorylation. You're adding a phosphorus. And then it's going to turn into adenosine triphosphate again. You just added a P. So if you want to make out another little side note just to explain your drawing, that it's ADP plus a P equals ATP. Whatever works for you. Alright then, we're going to start a new video for part 2 of enzymes because our video is getting a little big and it won't upload. So that next video will be on in just a second.